Hello, today Colleen and I will be discussing and comparing skeletal versus smooth muscle. First, I will briefly discuss the arrangement of skeletal muscle. It is very different from that of smooth muscle. This photo may look familiar to you. It was taken from Guyton and Hall's textbook of Medical Physiology, 12th edition. It shows the breakdown of skeletal muscle. When looked at closer, you can see that skeletal muscle is comprised of muscle fasciculose, which is a muscle bundle of a muscle fiber surrounded by connective tissue. When looked at even closer, this becomes a muscle fiber. Each muscle fiber is composed of bundles of myofibrils. Each myofibril has a thick and thin filament that form repeating dark and light bands called sarcomeres. Each sarcomere is comprised of 2 actin myofilaments and 1 myosin filament. The sarcomere is the functional unit of the myofibril and are separated by Z-discs. From Z-disc to Z-disc is 1 sarcomere, and more discussion will be on this later. The neuromuscular junction is on the muscle fiber. 1 nerve innervates 1 fiber. The repeating striated arrangement of the myofibril arises from the contractile filaments that compose the sarcomere, the thickened filaments and the thin filaments. The thick filaments, myosin, are the center of the sarcomere, also known as the A-band. Oops, sorry about that. The M-band is in the center of the A-band and links the thick filaments together. The thin filaments are about 1/2 the diameter of the thick filaments and are composed of proteins, actin, troponin, and tropomyosin. And there's more of this on Nagelhout and Plaus, page 785, if you're interested. The Z-line or Z-disc connect the thin filaments, or the actin, on either end. This less dense area is also referred to as the I-band. So I encourage you to look at this and get to know the different areas and what the letters are associated with, because it does come up and can get a little bit confusing. This next slide here, this is an image of the neuromuscular junction. We'll start with the image on the left. As you've learned previously, some stimulus causes the opening of voltage-gated channels, allowing the slow influx of sodium. This causes the nerve muscle to reach threshold from about negative 90 millivolts to about negative 65 millivolts. This results in a rapid-acting voltage-gated sodium channel to open, allowing even more sodium into the cell, raising electromotor force far above threshold, otherwise known as depolarization. High EMF activates voltage-gated potassium channels to open, allowing potassium to exit the cell and return it to its resting membrane potential. At this time, chloride also moves into the cell, allowing for hyperpolarization. The sodium-potassium ATPase pump reestablishes the sodium-potassium gradient. Meanwhile, the action potential that was generated by this ion movement into the cell travels down the nerve to the neuromuscular junction, causing a presynaptic influx of calcium into the nerve ending. As we all know, calcium in results in neurotransmitter out. The neurotransmitter at work at the neuromuscular junction is acetylcholine. Now focus more on the image on the right. In the presynaptic neuron, acetylcholine gravitates towards the dense bars, where it is then exocytosed into the synaptic cleft. 2 acetylcholine neurotransmitters bind to the alpha sites on the nicotinic acetylcholine receptor located at the synaptic cleft on the muscle fiber. This binding causes a conformational change of the receptor, allowing sodium to enter the skeletal muscle fiber, thereby creating a skeletal muscle fiber action potential. This action potential travels along the sarcolemma, or the skeletal muscle cell membrane, and deep into the muscle cell via T-tubules. T-tubules run parallel to the terminal cisternae located on either side. This is known as the triad, 1 T-tubule plus 2 terminal cisternae. As the action potential moves along the T-tubule, it stimulates the opening of the voltage-gated calcium channels on the sarcoplasmic reticulum through the help of the DHP receptor on the sarcolemma. That uncorks the ryanodine receptor on the sarcoplasmic reticulum, allowing for a massive release of calcium. This intracellular calcium is used within the troponin-tropomyosin complex to cause a muscular contraction.
Okay, so as you can see in this picture, calcium binds with troponin C on the troponin-tropomyosin complex. Troponin I binds with the actin and troponin T binds with tropomyosin, T for tropomyosin. When calcium binds to tropomyosin C, it exposes the actin binding site. Myosin has a high affinity for actin binding sites. ADP on the myosin head causes a cocking to 90 degrees, or also called a power stroke, and ADP cleaves into ATP, which causes the release of the head from the binding site. This occurs in the presence of calcium after an action potential and is the basic mechanic behind skeletal muscle contraction, a 2D movement. Smooth muscle can be stimulated 4 separate ways: by humoral factors, electrical means, chemical mechanisms, and mechanical stimulation. There are 2 types of smooth muscle: unitary, located in your gut, and multi-unit in the eye and piloerector muscles of hairs. In the multi-unit, each fiber is innervated by one nerve and operates independently. Outer surface of each fiber is covered in collagenous glycoprotein to protect it from other nerve impulses to allow very refined movement. Each fiber operates independently. Unitary muscles is a mass of fiber that functions as a single fiber. They work in a syncytium via gap junctions. This type of muscle is found in the gut, vasculature, ureters, bile duct, etc. As you can see in the picture above, actin and myosin are at a 15:1 ratio. Actin connects to dense bodies. Side polar cross bridges allow myosin to pull actin filaments in one direction on one side while simultaneously pulling another actin bundle in another direction. This is what allows smooth muscle to contract as much as 80% of its length in a 3D fashion. Skeletal muscle can only contract about 30% of its length. This image demonstrates the inner workings of smooth muscle contraction. It varies from skeletal muscle. As stated earlier, smooth muscle contraction is stimulated by mechanical, chemical, electrical, or humoral factors. After an action potential opens the voltage-gated calcium channels, allowing calcium into the cell, which enters a positive feedback loop with the poorly developed sarcoplasmic reticulum, which in turn also releases calcium. This intracellular calcium binds to calmodulin, forming a calcium-calmodulin complex. This triggers and activates myosin light chain kinase, which in turn causes the myosin light chain to phosphorylate and bind to actin, resulting in a contraction. Myosin and actin bind for long periods of time, known as the latch mechanism, and this is done so without much oxygen or energy consumption. This is a huge difference between smooth and skeletal muscle. This is a rough overview of what is known about smooth muscle contraction.
What we provided here is a brief chart that will compare skeletal to smooth muscle. It's a common question that will come up, and they do vary greatly between the two, so it's a concept that you should try to put a good handle on. Feel free to add on to this chart, to take it and use it as your own. It's just something that we came up with in hopes of giving you a good comparison between the two. I hope you enjoyed this video. Thank you for watching.